Hey guys, welcome back to my channel. In this video, I'm going to be going over IB Biology topic 10.2, Dihybrid Crosses. If you guys are new here, I'm basically a small-time YouTuber. I've recently just started and I only have about 30 subscribers, so it's a growing channel. If you guys subscribe, I'll really appreciate it. You guys don't have to, you guys can just watch the video and leave, but if you do subscribe, that will mean a huge lot to me. And leave any comments down below for any video suggestions you have, and it's almost guaranteed that I will do it because I don't get many comments. Anyways, let's get right into the video. So a dihybrid cross is concerned with what we call linked genes. So linked genes are genes on the same exact chromosome. Because they're on the same chromosome, during meiosis, two of these genes are going to be inherited together. As in, we don't split the chromosomes themselves, we don't cut them in meiosis. So the cell which this green gene ends up in the final stage of meiosis, let's say a sperm cell, well, we can be guaranteed that this yellow gene is also going to be going with it. So we have linked genes. Previously, we were dealing with unlinked genes, which followed the law of independent assortment. So unlinked genes are genes on separate chromosomes and different gene loci. So if you have two different chromosomes and two sets of genes, let's say yellow here and green here, when they align during metaphase stage of meiosis, they can align randomly with, let's say, yellow one on this side and green on this side. Maybe yellow and green are both on the same side. You never know. It's random, so it's not dependent on each other. There's no rule saying like if this yellow one goes to sperm cell A, this green one will go to sperm cell A as well. It could, but there's no rule linking those two together. So we call them unlinked genes. And because of that, they follow the law of independent assortment, which simply states that pairs of alleles that are on different gene loci are inherited independently of one another. So one's inheritance does not affect the other, and these genes are called unlinked genes. Anyways, unlinked genes had what we call one of those Punnett squares, and that was a monohybrid Punnett square. So it was a 2x2 Punnett square, which was quite simple compared to linked genes where we do dihybrid crosses, and we have an 8x8, 4x4 Punnett square. So yes, Punnett squares can get even harder. When we're doing dihybrid crosses, we get the genotype of the organism, and we have to first deduce what type of gametes will be formed. So a female will form secondary oocytes or ovum or eggs as the gamete, and males will produce sperm. This is in the case of humans, of course, and some other animals and plants, I guess, where plants have pollen and ovules, so that's a different story. So how do we determine these gametes, you may ask? Well, what we need to do is do something called FOIL. This might ring a bell for you guys who've done quadratics in maths, where we go first, let me do a different color, first, outside, inside, last. So that literally means we multiply, or you could think of this as multiplication. We get the first parts of these two, and that's one gamete. We get the outside parts of these two, that's the second gamete. The inside parts, that's the third gamete. And the last parts, that's the fourth gamete. They're not in that order, but those are the four different types of gametes that can be created. Gametes are haploids, so another parent could have, like, so let's say we're working with this one, they could have big G, big H, and that could be the other haploid cell that this one combines with. But we have half the genes. So for any given gene, we should have two alleles, but we have one because it's haploid, and the other gene will be influenced by the complementary gamete. So if a sperm, it's an egg, and if it's an egg, it's a sperm. You get the idea. Say we have another organism, this time little g, little h, little h. Well, if we use the FOIL method each time, if we get first, outside, inside, last, we're only going to get GH. So that's the only important ratio in the cross, right? So sometimes you don't have to have 4 because you're going to get GH, 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 GH. So it's going to be the same one, so we can simplify that to 1 because all the gametes will have the same exact genotype. Now let's have a look at this example I found from BioNinja. In sheep, an allele for black wool, B, is dominant over the allele for white wool. Similarly, the allele for horns is dominant over the allele for being hornless. So first we identify which one's which. Okay. Pure breeding horn sheep with black wool were crossed with pure breeding hornless sheep with white wool. State the genotype and phenotype of F1 individuals and produced as a result of this cross. Okay, so let's look at keywords. The most important keyword here is going to be pure. Pure breeding. That's kind of hard to see. But pure breeding black wool and pure breeding horn sheep with black wool means that they're all going to be homozygous for each of their traits. So if horn sheep is big H, then this sheep is going to be big H, big H. But let's do the wool color first. So it's a black sheep, so it's going to be big B, because it's going to be the same. It could be this one, it would have the same phenotype because this is still going to express the dominant trait. But we have this big B, homozygous dominant trait because it says pure breeding. Next we look at this one. This sheep is pure breeding hornless and white, so that's little b, big H, little h, little h. So those are going to be the genotypes of the parent organisms. Now we need to work out the gametes. I'm going to write G for gametes. So if we use the FOIL method, like it's one of those examples where we literally only get BH and BH. Those are the only options we could get. When we're making a Punnett square, we have to make sure that we label the phenotypes that are present. So we have big B, little h. Big B, big H. It would probably be easier to like this. And little b, little h. So those are the genotypes that are available. I think I accidentally said phenotypes earlier. I meant genotypes. And we simply put these together. So we have big B, big H, little h. That's what we have as our genotype, and we have to identify the phenotype as the as per question, and we get black horned because horned is dominant. We have the dominant. Heterozygous dominant means dominant is expressed, and heterozygous dominant for B as well means the wool is going to be black. That's part A done, and let's move on to part B. So two F1 generation, F1 offspring were mated. Calculate the expected ratio of phenotypes in F2 generation. Okay, so going off memory, the F1 generation was guaranteed to be big B, big H, little h, right? So that's going to be the organism's genotype. But we need to work out the gametes, so we have to use the FOIL method here. So first, outside, inside, and last. That's going to be the F1 generation's guaranteed or possible set of 4 phenotypes. 4 genotypes for the gametes. So if we have 2 F1 offspring mated together, that means we have to label it like this. So we get BH, oops, I have to go here, clumsy, BH, BH, So we're essentially drawing out like the possible gametes that can combine. So we can get big B, big BH combined. It would obviously be this. I'm just gonna complete this table off camera and I'll come back to you guys. But these are gonna be the genotypes that are present. So in this question we're asked to write out the expected ratio of phenotypes, right? So the phenotypes possible are black horned, white horned, black unhorned, and white unhorned. And let's see, I'm going to count the number of each one. So we have black horned, we have this one, this one, this one. All of this has to be black horned. All of this has to be black horned. And is there another one black horned? This one's black horned. This one's black horned. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9. I don't think there are any other black horns, so we have 9 black horns. Let me draw that in a brighter color. 9. Let's see, white horn. We have white horn, white horn, white horn. So 3 white horn. Black unhorned, we have this one, and this one. So 3. And white unhorned, we have 1. So we get a 9 to 3 to 3 to 1. And this ratio is one of those magical ratios. It's not the golden ratio, but it's one of those special ratios that are important for dihybrid crosses with heterozygous for both traits. So if you ever cross two things that are heterozygous for both traits, so like big B, little b, big H, little h, and you have that for both, you're always going to get a 9 to 3 to 3 to 1 ratio between the both being dominant, one recessive, one dominant, one recessive, and recessive. So that's going to be a set ratio that you're going to have to memorize, because sometimes in paper 1 you won't have time to draw out this whole thing. But if you can, I do recommend drawing it out just for certainty. And also, also in paper 2s, make sure you draw out the Punnett square, because that can have mark points, so don't just memorize this ratio. There are also other ratios you need to consider. I might make a new video on that, but in the meantime, that's going to be all for this video. Hope you guys learned something new in IBB Biology 10.2, and like I said earlier, this YouTube channel is quite new, and I wanted to grow, so if you guys could leave a like, subscribe. You don't have to, you're under no obligation, but if you did, that would mean a lot to me. And if you leave any comments about video suggestions or any topics you want me to cover, I'll be happy to do that for you. So that's going to be it for this video, guys. Hope you guys enjoyed. This is Latify signing out.